Okay, welcome back. No major casualties, no people who have dropped out. Well, everyone happy and back, wanting to know more. For those of you, and I know there are a few in the audience who are catalyst nerds, I can say that, I mean, this is a fairly detailed paper. This is an outer sphere mechanism as compared to inner sphere. I don't want to go into what the difference is, but for those of you who are more working as professional catalytic chemists, this might be of interest. So, moving on, is this better now? Do you hear my voice through the microphone? It's okay? No complaints? Good? My voice is still operative, so I feel pretty okay. So I think I can go on for a couple of more hours. This is a list of criteria that I connect with efficiency in manufacturing. Simon might have a different view, but over the years, these are some of the attributes that I connect with if a process is efficient. The very last slides that I have in this package, I always thought, how should I end this long lecture? What should I say? Thank you, or should I say something else? I will show you the most efficient process that I've been involved with developing. So I'll keep you in suspense for what that process might be and why is it so elegant. So stay to the end, otherwise you won't know. That's a teaser. Short, some of these things are so trivial I don't need even to comment on them. You as chemists will understand this. People who are not in chemistry might need an explanation. Convergency, catalysis. We now know that catalysis is on the forefront of green chemistry. So working in catalysis is rewarding. It's the right thing to do. Doesn't mean that everyone has to work in catalysis, but there are so many catalytic processes out there. I think colleagues here in industry have to correct me if I'm wrong. 90% of all chemicals that are manufactured, I would say, are done via catalytic processes. Would anyone challenge that? I think it's a big majority of processes are done one way or another where catalysts are involved. That's the message. So again, working as a catalytic scientist is right. Our chemical industry relies on good catalytic processes. Atom efficiency, I will come back to this when I talk about sustainability, but having a lot of protective groups, it increases weight. You take them off, they don't add to the final molecule, they are just there because the chemistry is insufficiently well developed. You should try to get rid of all protective groups if possible. Telescoping, this is more a commercial or a commercial large-scale manufacturing technique where you try not to isolate each and every stage, but you run them in one sequence and then, or in situ chemistry if you wish, and then you work it up at the end. Every workup takes time and you're bound to lose yield there. So by telescoping, it's a quicker, a faster process and a better outcome as in yield terms. Don't change solvents too often. Again, in the lab, you as lab chemists, you have a rotor evaporator. You put your round bottom flask at the evaporator, you go for a coffee, you go to the restroom, and when you come back, the solvent is gone. In a factory setting, even in a pilot plant, it could take a day to take out the solvent from a 1,000-liter reactor. And then you wouldn't even be sure that it's all dry. Evaporate to dryness, what is that? Yes, you can do it in the lab. You can hardly do it in large scale. So by changing from one solvent to another, again, it takes time, something you should try to avoid. Sometimes you have to swap solvents, but try to avoid it. You always need to demonstrate how does your chemistry work in water. Because again, going back to the literature, you say it was run under dry conditions or water-free or something. Yes, again, in the lab you can do it with meticulous control of the water content, but in the large scale you cannot. There will always be 0.1% of water left, which doesn't sound as a major deal, but considering that water is a light element, so even small amounts of water amount to very many moles. 1 kilogram of water is quite a number of moles, as you know, 56 or so moles. So if you have a reaction going on where you have maybe 200 moles of both reactants, 200 each, this might destroy. And we have come across this. The water is a tricky element to take care of in your processes. Purification, I already mentioned that crystallization is our workhorse. We always go for finding a crystallization system that can do the purification. If you find one, it's miraculously how effective they can be. But extraction combined with crystallization will often solve your problems. Distillation, I've hardly come across any process. There were a few ones, but otherwise distillation, the high temperature, at least normal distillations, sensitive molecules, a lot of the material is bound to degrade once you, again, in a small experiment, you will distill it fairly quickly, but if you have a 1,000-liter reaction solution that you need to distill, it takes hours and hours, and during these hours a lot of your precious material will degrade, will be destroyed. So nothing to recommend. Environmentally concerned goes without saying. We cannot develop any process today without documenting the environmental impacts. We can always claim we are doing small amounts, so what's the big deal? We are not producing many tons of material, but that isn't enough. We have an image problem if we are not concerned about the environment. Our management needs to go out to the shareholders, to the public, and say, we follow environmental legislation, we are operating according to the best principles when it comes to protecting the environment. And so this has to be taken into account when you develop your processes. Scalable, obviously the technologies you're using have to be scalable. They should not just work in a lab scale, but they should work on 1,000 liter, on 4,000 liter, maybe even larger than that. And all technology isn't linearly scalable. So you have to figure out, can I do this kind of chemistry on this sort of scale? Robustness. Of course, you need to ensure, if something is registered as a drug, you need to ensure to the market that you can deliver the material. There should be no shortage of a medicine. It still happens for various reasons. But unless you have a robust process, meaning a process that delivers the expected quality, the correct compound, and the correct yield, give and take, you have no control, and this means sometimes you will see an impurity profile that is outside the specifications. What to do now? How do we handle the situation? So robustness is something you can build into your development. Statistical means of showing the variation over time. This is monitored very, very carefully these days in order to avoid problems with in unrobust performance. It has to be safe, as I already told you. Chemistry is dangerous in the wrong hands. So you have to ensure. The problem is that we in the lab, we know how to deal with this. We are trained, we know what could go wrong. For people in a factory setting, they are not chemists per se. They follow given instructions. So you have to ensure that those people on a different educational basis than yourself can handle the process without risking their lives, the equipment, the environment, what have you. So this safety aspect and making it intrinsically safe. So this is again something you are not, when you have developed your process now, well, we need to devote some resource to ensure that the process is safe. That's wrong. That is the way it was done before, and you will inevitably run into surprises. You need to build in the safety aspect from day one. When you conceive the process, when you see it before yourself in your brain, in your head, you need to think, this has to be safe. Is it safe? Can we run this at the given scale, etc.? Freedom to operate, this goes to what I said earlier. If there is a patented methodology, or if your sequence takes you through an intermediate that somebody else has patented, then you're in trouble. You could solve it in two ways. You could ask for a license and pay for a license, and it sits in the owner of the IP to say, I give you the license, but you have to pay this much, or no, you can't because I don't like you, or you are a competitor. I don't want to give you any advantages over what we are doing. There is no rule that says you have to give someone, a third party, a license of your technology. It's yours, you own it. Once the patent lapses, then it's free for everybody. But during the time the patent exists, you cannot abuse this. We expect third parties to accept and acknowledge our patents. We have to do the same. I have a case later on where one of our synthetic routes actually went through a patented intermediate, and I'll show you how we solved that. The other option is obviously to try to come up with a new technology or a new chemical route where you won't hit any patent problems. This is important. Something you have to start thinking about very early in the work, so that you're not developing a process that will have these problems. And finally, cost conscious. Drugs are fine chemicals. They are advanced molecules, many of them even extremely advanced. They are costly, but you need to produce them at an acceptable cost. No one can define what an acceptable cost is, but that's another issue. But you should feel satisfied with yourself that we have done all we can to reduce the price as much as possible. Pharma manufacturing, a few facts. This is a fine chemicals industry. So the volumes are small in a global comparison. A few kilogram high potency rare disease drugs might suffice for the demand in the whole world. A few kilograms. This is lab work, more or less. Up to several hundred tons. Antibiotics, these are relatively low potency, high dose, NSAIDs, non-steroidal anti-inflammatory drugs like naproxen, where you take quite large doses. Maybe the annual production of antibiotics and NSAIDs, a couple of thousand tons, but that's the limit. Looking at how many petrochemicals are produced, we talk about millions of tons. Acetic acid, sulfuric acid. This is a different ballgame. There is a very interesting relationship. I'll show it. I didn't bring that slide. Is there something to write with on the... Wonderful. I haven't used these for ages. Here. It's a diagram. Like this. Where we have volume here. You can say tons per annum. And you have cost. These could well be logarithmic scale. So how does this curve show the interdependence? It's like this. So if we have, let's say, log 10 million tons is about the largest commercial, let's go for that. And obviously here is 0. Cost, let's make it a 4, and then it varies in arbitrary currency, doesn't matter. So what does this show? Huge volumes at low cost, or very, I mean, here we're talking about $1 per kilo. You can buy a lot for $1 per kilo, believe it or not. What do you find in this area? Commodity chemicals, sodium chloride, sulfuric acid, acetic acid, all these chemicals that you find all around you at virtually no cost at all. Elastomers, you go up a little more sophisticated monomers, you will escalate on this curve. Intermediates could be somewhere in this region, where you talk about, this is more advanced type of compounds. $50, $20, somewhere in that range, maybe upwards, depending again, complexity. When you reach this part, what do you find here? Of course, pharmaceuticals. Small volumes, a couple of hundred tons at most, at very high prices. You will find fragrances, all sort of, well, chemicals with a specific use, more sophisticated use, directed use. So the sodium chloride that we're talking about here, sulfuric acid, you will use across the whole industry. The more specific the use gets, the more expensive, the more sophisticated the molecules. This curve could obviously be extrapolated even further now with the advent of the biomolecules, the proteins, the antibodies. That's sky is the limit. you could easily pay, well, it's approaching $1 million a yearly treatment with one of those most sophisticated antibodies. Then we get into another discussion, how much should healthcare cost? I don't have the answer, and I don't intend to go into debate about that. Everyone will have his or her own take on this. But this is, it's useful to know that large volumes, often very, very low prices. So here, the process chemists working with products in this range, they have to work their butts out to really gain a tenth of a dollar or a cent even to improve. They trim the reactions, and it could be a question of staying alive as an industry or being killed because someone has come up with an improvement that makes it 1 cent cheaper, and everyone will buy it cheaper because it's a commodity. Just buy it as cheaply as possible. Here we talk about unique products that you might be the only one who produces. So here people are much, much more willing to pay the extra charge, but approaching to this end, they won't. So it's you operate in totally different domains of this curve. This is what we need in the R&D stages, 10 to 100 kilograms. So that's what we use our pilot plants for. Production for the early clinical trials, low kilogram amounts, up to a couple of hundred in the late phases. Batch mode is still the way materials are produced in the pharmaceutical industry, meaning you add everything to a reactor, you run the reaction, you work it up, or you just clean it and then move on to another reactor, run the next stage, etc. So batch mode in relatively small units, 4 to 6 cubic meters. In fermentation, you have to multiply by 10, if not more. There are even reactors that are a couple of 100 cubic meters, but not in the synthetic chemistry space. Flow chemistry is something that we see more and more of. People have overcome the fright to do continuous chemistry when it comes to manufacturing active ingredients. I will talk about that later. The whole industry of making pharmaceuticals is embedded in a very, very strict legal framework called GMP, Good Manufacturing Practices. It is exactly what it says. The patient who sits out here somewhere has to rely on that the quality that is produced is fit for purpose. No tampering, no contamination. I will show a few cases where this has happened. This happens all the time. It happens while I'm standing here. There are so many ruthless people out there who just want to make money. So they offer adulterated compounds. You probably come across this. It's an increasing problem rather than something that is about to disappear. So they offer you something that they claim is 99% pure, and it's nowhere near that. It might even lack the active ingredient, sugar or flour or whatever have you in there, or a totally different active ingredient. Might be dangerous for your health rather than the other way around. So in the beginning, everyone felt that these GMP rules are just a nuisance. Takes so much effort, it's so costly, but when you've worked in the business for a while, you realize this is a must. We need these rules, and I will talk a bit more about what they entail. This is a trend that has been with us for, at least in my company, 15, 20 years maybe. that you try to offload the burden of keeping big factory units running for making chemicals. It's not the chemical per se where we will make the difference, it's the medicine and its performance. So you could outsource a lot of this work to more chemically oriented companies. A pharmaceutical company is per se not a chemical company. They have different agendas and capabilities. In AstraZeneca, this was operated to a crazy level actually, where every internal manufacturing capability was closed and outsourced to Eastern countries. And I'm talking about China and India, only to realize some years later that also they can have problems. They can have delivery hiccups, they can have quality problems, etc. So the trend now, the pendulum swings from one extreme back a bit more into using the own facilities. The problem with having manufacturing capability in the pharma industry is that so few products succeed to go to market. So you could sit with assets, production assets that cost millions and millions of dollars, and you have nothing to do there because you haven't produced a new medicine. The purpose that they were built for, most of the plants are built for particular purposes, for a particular process, so you cannot just swap them around. And even if you could swap, you have to prove that there is no cross-contamination, which is again is a GMP rule. You have to show the quality, etc., etc. So building very costly factory units and not using them from a financial point of view, this is madness. Yet you need to have these facilities ready before the product is registered. You have to do all the investments, a couple of $100 million to build a new factory, before you know that it will get registered. So you can see in a nutshell that this is a difficult equation to solve. Another good question, I think, is whether scale-up is trivial. For some people, if a molecule has been made in the lab, then it's been made. So what's the deal? Just make 1,000 times or 10,000 times more. Okay, bye. It depends. Do you have experience from similar chemistry? How good does your process have to be? Is it a small volume? Is it a kilogram quantity? Is it 100 tons? Obviously the demands will be quite different. You can live with a mediocre process for making a few kilograms, but making 100 tons, you will probably be more diligent. How pure does it have to be? Are there any patents that forces you to go in a certain direction? What timelines? How much time do you have? Time is very precious and you don't have enough of it. How long are you supposed to work until you have a solution? Until you can deliver? That's the reason I showed you in the omeprazole case. We had to revert to chromatography to satisfy the needs of the pharmacologists and the formulators. What is different to running in the laboratory? The risk factor, the volumes are much higher. If something goes wrong in the lab, maybe only you will be affected as the chemist in charge, but if a factory blows up, it might be different. It might destroy quite a lot, the whole site or even more. The large equipment is great from many aspects, but it's much more inert. It takes longer time to heat something, to cool it down, especially if something goes wrong. What you do as a chemist is try to cool it quickly because temperature raises the reaction rate, etc. Cool it down. to cool down a reactor at 150 degrees down to room temperature could take hours. In the meantime, you better leave as quickly as possible because there is no point of return. Stirring is one of these unit operations that seems very trivial. In small scale, you can see it stirs around, it mixes well. But on large scale, it doesn't. And that's the reason you have hotspots and you can have deposits on the walls of the reactors, etc. Addition of reagents, something is added momentaneously. You do that in the lab, you just pour it in, it's added. Here it is bound to take longer. Separation times, separating phases could take hours, could take days. So what you as a development chemist need to do is to check out all these parameters in the lab. You have to know that separation could take several days, even if it in the lab goes within 15-20 minutes. Do you know what is are the phases stable enough so that you won't degrade your product if you have to leave it standing for 2 days? This is something you need to know. If you don't, you are bound to run into surprises when you scale up. And then obviously cross-contamination again. If you run in multi-purpose plants, as we are all brought up to do because of the investment cost to build equipment only for a given process, you need to show that there is no contamination between different batches, which might be quality affecting. The design principles, we heard about those in part already. Shorter routes, low yielding steps early. I guess that if you think strategically, that's the right way of doing it, because if you have it late on, then you have brought a lot of material. You have carried a lot of material through your process only to lose a large portion of it at the very end when the goal post is very, very near. So low yielding steps in the beginning. Expensive reagent that goes without saying, as late as possible. They are expensive, so if you use them in the first step, you have to convert a lot of material. In the late stage, it's less. High dilution steps late if possible, because it adds to the volume. You need bigger reactors in high dilution. And this is something that is often forgotten. People are so hung up about the early chemistry, and they forget that it's the late stages where the quality of the product is decided. Make sure you control the quality in the last steps rather than putting in all sorts of efforts in the beginning. You can always, if it's a lengthy process, you will find ways of purifying material that isn't entirely perfect from the beginning. Starting materials, what is available on the market? This was a compound we were after, and it was actually prepared in small lots in London. Is this reliable? Does this company have the capability to deliver a ton if you need? Most likely not. A good example I can show you that hit a project that was high profile and very important is this one. A simple amino acid, acetylene carboxylic acid, an analog to proline. We needed this for a high-profile project. It's a beta-turn amino acid, so it will impose a certain conformation to a cardiovascular drug that we were operating with at the time. The discovery chemists said it's commercially available. Perfect. So we will always have access to this amount when we make the larger quantities. When we started the project and looked around, it turned up that lab quantities are available. 100-gram bottles, something we needed 10, 20 kilograms, something like that. So how can we do this? We put external supply professionals onto the track. Can you find this material somewhere in the world? This is a natural amino acid. You find it in certain lily flowers, so you can isolate it actually from a natural source. And this is what we found through this agent. It was a French company that had manufactured this via chromatography, very time and costly process. They had, how much did they have in their store? They had isolated from sugar beets, a very nasty process, and they refused to repeat it. They said, you can buy whatever we have in the store in France, 100 kilograms, something of that order, and we paid more than gold for it, I can tell you, but it was so precious for us. So we bought everything they had. That was what was available. So instead of focusing on the downstream chemistry starting from this material, we had to devote quite large resources coming up with the process of making it instead of relying on others to have it ready for us already. This just shows in a nutshell, you need to follow up. If someone says this is commercially available, okay, great. On what scale? How much? Is it reliable? That's the reason I'm telling you that going from lab to large scale often gives you questions that you never thought you would have to answer or ask in the first place. Hazards. This is a very striking picture from biocrop science. Maybe you know of this accident.
I think it's alcohol preparation.
Yeah.
It's insecticide.
Yes. Yeah. Yeah.
It's accumulation of toxic explosive intermediate, very energetic.
Yeah.
That bursts.
Yeah. Yeah. And you see the results here and the fatalities here, and it's only 10 years ago. What has happened to this site now that we have an insider here?
And I think it has been revealed.
Okay.
Alcohol is less and less used, but I think everything has been.
So was the reason the human factor, or was it a mistake, or?
I don't remember why there was accumulation of this. Is it technical or human? I would say technical, but I'm not sure.
Yeah. Okay. Well, you will all have seen pictures like this. They are abundantly available. This statement speaks for itself. A lot of injury can occur. Why do these happen? I think, and you have to correct me if I'm wrong, you don't understand the chemistry or the thermochemistry. Temperature is a trigger for these events. High temperature could unleash reactions that you are in no control of. Maybe the engineering committed mistakes. Yeah, oops. You didn't design it properly for the heat transfer that you need. There are enormous amounts of heat generated in large reactors. How do you deal with this heat? It stays in the reactor. You can't just evaporate it off. How do you deal with that? Control systems, safety backup systems, in case something goes wrong. What do we do? How can we, let's say, reduce the risk or turn it to, or put it into a space where there is no risk anymore, by temperature reduction or by adding a cooling liquid or maybe water to stop the reaction immediately. I mean, you need to be prepared for these kind of events. Operational procedures and, importantly, training. People need to know how to act if something goes wrong. Start training activities after the accident. That's also important, but you need to be more proactive than reactive here. What you often do in the lab, and I don't blame anyone for it, but you have all been victims to this. You solvent, reactants, low temperature, then you heat it. And when you get back or you do something else, then the reaction is done with and you work it up. On the large scale, this might put you in a position where you lose your control. What you could do instead to mitigate this, add the reactant slowly at a high temperature, which ensures a fast conversion, so you never accumulate potentially reactive starting materials. Or move into a continuous flow, because at every instance there will be very little material reacting. It's instead a flow system where segment after segment undergoes the reaction and then you quench it when it's already done with. So you never have large amounts of potentially dangerous material in your reactor. And that's one of the drivers for actually, especially for more energetic type chemistry, for moving to a flow setting. So these are pictures. So from laboratory, where it's local, to factories and to real big blasts with several casualties and harmed people. You can always ask whether it is safe, and is this always unsafe? But the take-home message is that you need much more experimental evidence, understanding of your chemistry when you move along this arrow. The bigger the scale, you need to know much more in detail what might happen. You don't want to see these kind of situations in your neighborhood. I can only mention the Bhopal accident. You're all aware of something got totally wrong. And then you can, and this was operated, the plant was operated by one of the foremost chemical companies in the world, yet it had this catastrophic effect. So this is to illustrate the two scenarios, how you should run reactors. In the lab is a controlled environment, operated by specialists. You have more freedom to design it a bit as you feel fit, and you don't need to overcomplicate things. This reaction, you charge them all in batch reaction. You have everything in your reactor. You heat it, it's over in an hour. Sure. But what you should be doing in the large scale is to charge the solvent and one of the reactants, heat it to 75, and then start, because the reaction will not happen until you charge the other reactant, the other substrate. And you do it in a controlled manner. If something goes over the top, you can always stop charging. But if it's already in there, you're lost. You have to kill it all by either cooling it down quickly or just let it explode. It's nothing more you can do. Neat reaction, high concentration, limited capacity. The temperature, the adiabatic temperature increase will be much higher. You might see decomposition, large excess of reagent in method 1, favor secondary reactions. Yes, could be. You have to check this in your particular case. All reactions are difficult to control when you have everything already charged to the reactor. And as I just mentioned, slow addition gives you that fine control that you need to stop the heat generation. Stop addition, it will inevitably calm down the reaction. How much more time? We still have some time? Yeah? Okay, good. So, now more of synthesis. And the subheading, designing the best route. I guess you've died to hear about what is the best route. Any questions about what I talked up to now? Starting to feel that you need to replenish your energy reservoirs, or yeah, it's a bit brain damage, too much information. Well, we will finish for lunch soon. I'll just start this chapter. So looking at it, what I described from a process chemist's point of view, when we start the projects, we know a few things. This was described by Simon yesterday. We know that the molecule has been made. We know how it was made. If it's a clever chemist in discovery, he or she might even have ideas for improvements. Some physical data, some spectral data. What we don't know in most cases is what does the intellectual property situation look like? Are we blocked? You can always, and this is a very interesting question, so I think it's good to mention it. When in the development of a new medicine do you have to take third parties' patent rights into account? It sounds as a very trivial, simple question, but if you think about it, it isn't. You know what patents is there for? It's to prevent that you misuse or abuse third parties' rights to inventions. So whilst you are developing something, when do you hit the point where your activities will be able to be seen as commercial? Because it's supporting a commercial endeavor. We do this for a reason. We develop new medicines for a reason. Not ignoring the fact that the patient is the target, but still we do it for a commercial reason, meaning we want to register a new compound, a new drug, and get our investments back with some profit. That is, in easy terms, the way. So when does a project become commercial? It's not so clear. And I think different individuals might have different views. My view is that in the discovery phase, you are still looking. You haven't decided until late in the discovery which molecule to go for. We heard that very well described yesterday. But once you have picked a specific molecule, this is what we will develop. Then you start to move into a gray zone where you might say, we do this further development, preparing for clinical trials, for documentation, etc., for economical reasons at the end. So I think an ethical answer to this is to try to be mindful of the patent situation the very day when a compound is chosen as candidate drug, because then you have made a choice. You can point it out, and you will now invest money to develop this to a final medicine. Then there is again a gray zone. When do you really have the data to say that the likelihood that we succeed is above a certain percentage, or we feel it's time. The scale of manufacture, for example, making the molecule in the pilot plant, that's when it becomes commercial, pseudo-commercial. We don't own any money, but we do it in order to become commercial. So I would say all the early stages, don't bother too much about the patent situation. You know, only very few molecules will be selected anyway. But when you start development, you need to, and this is again case by case, you need to reflect on what feels right for me, and start to examine the patent situation, and if need be, ask for a license or try to come up with something different. These are the options you have. So if we know these aspects due to the work of Simon and his colleagues, there is quite a lot we don't know. We don't know the best synthetic route. There is seldom a medicinal chemist who says, this is the best way. It might be the way that we go forward with anyway, but they cannot claim that they know this. Cost of goods, scalability, bulk availability of starting materials, we have already mentioned this. This is an interesting one. Which is the best salt? This sounds again very trivial. The FDA, to my knowledge, lists about 50 counter ions that you can use for making a salt. Very rarely you will use the drug as the free base for various reasons, stability, formulation work, etc. So you do a salt, and it doesn't always have to be hydrochloride. 50 different counter ions. Either it's a base or it's an acidic molecule. There is a good choice here. And then polymorphs, the same molecular setup, but a different crystal lattice. And why is this important? Because the bioavailability differs from one polymorph to another. Might differ, it's not clear, could differ. But there are cases where companies have come up with better polymorphs. They are more soluble, they are more stable, etc., etc. So one of the last things we did in AstraZeneca at the site I operated in Sweden, that was to build a robot for polymorph screening, because you have to patent your polymorphs, because previously at least it was possible to patent a new polymorph of a known drug that somebody else had invented, and that's not what you like to see. So you need to know all the polymorphs that are out there. And then it doesn't stop here. Should it be a hydrate? Should it be a solvate? These molecules have a weird tendency to appear in different shapes and form. Should it be a hydrate? Should it be a monohydrate, a trihydrate? Should it be a solvate with ethyl acetate? You don't know. This is one of the major activities. How do you purify your materials? Where do you purify them in a process? And then what quality criteria? How pure does your material have to be? Does it have to be 99.9%? I got the question, what EEs, what enantiomeric purity does a drug have to have? You have heard my response previously. It depends. It depends. You cannot give a fixed It depends on what the antipode is. If it's inactive antipode, 1-2% would probably be okay. If it's an active antipode that even might be toxic, in some instances, you might have to purify above 99% EE. You can purify up to 100%, but the cost curve goes like up like this. Quality comes with a cost, and so you as a customer, as a sponsor of such a project, you must decide, is it worth it? Does it really improve the performance of my drug if I purify it to 100%? I would say in 99.999 cases it doesn't, but you can never be sure. PGIs, I will talk a bit about later, I think. Potential genotoxic impurities. So there are lists that regulators have provided for known infamous impurities that have proven genotoxic effects, and they will chase you. You have to prove that those are not present, down to sub-ppm levels even. So you can imagine, how do you ensure that your process, if those compounds are potentially present in your product, how do you do this? How do you ensure to a regulator it's clean? Cleanness is a very broad definition, but a very imprecise definition. What is clean? It's a very interesting aspect. I've finished with this one, sorry. So, quickly, the focus in medicinal as well as in process chemistry. This is from my perspective. So medicinal chemistry, you have to correct me if I'm wrong, from where you sit, Simon. Late stage functionalizations. This is strategically an important approach to making molecules. You have a common intermediate, not too complicated, so it allows you to make late stage functionalization when you build up your libraries. Everyone can probably buy into this. You can use various techniques, CH activation, biocatalysis, photoelectrochemistry, these kind of technologies that are just there to support making new molecules for a rapid assessment. Process chemistry, you heard it before, catalytic chemistry. When I started once, catalysis wasn't very big, and we were spoiled in the pharma industry then because we could charge, because we work with patented compounds, there was no competition. We could charge more or less anything for a drug. We were never challenged by not to the extent we are today, by the authorities. You have to drive down the price. So we could charge more or less, and also mindful that the active ingredient, the molecule, just contributes maybe 10% to the final cost of a drug, 10, 15, maybe just 5. So it's a small proportion. Why should I work the heck off and find catalytic processes? No one cares, not in the early days at least. So screening to find the best catalysts, catalytic predictions. We have quite sophisticated, AstraZeneca still has invested in a catalytic prediction software where you can say which ligand will work the best for this particular based on literature precedents, etc. So instead of screening 50 ligands, you can screen 5. It saves a lot of effort. Biocatalysis, I hammer in the message, it's coming very strongly. So biocatalysis you will see much more of in the pharma industry. Cross-coupling reactions. Suzuki is a workhorse. Heck, to some extent, not that much. Buchwald, we were one of the pioneers scaling up Buchwald chemistry. So when I met Steve Buchwald 15 years ago, I gave a talk on a project where we had scaled up to 1,000 liters or something, and he was enthusiastic because this gave him a very good argument for applying for new research funding, because he had only published lab work. And if you can demonstrate that there is an industrial use for your technology, you will get more money, or you get the money easier. So he was pleased. Molecular frameworks. I will show you some heterocyclic motifs, and this is all with the library work. People make crazy, people meaning the discovery chemists, make all sorts of crazy heterocycles, and it's for us then to find methods to produce those on large scale. Intensification of processes, meaning that not scaling to bigger sizes, but rather concentrating and making it more intense, getting more material delivered from smaller volumes. And this obviously entails the use of flow chemistry, continuous manufacturing, and sustainability. We have to live with green chemistry principles. I spoke to Paul Anastas 2 years ago, maybe something, and he was quite adamant about, he's the father, as you know, of green chemistry, the 12 rules, etc. He saw before him the day when we're not talking about green chemistry, because everything we do is green. You shouldn't go into the lab one day, say, today I will develop a green process, tomorrow I don't care. You should always be mindful that it has to meet the highest environmental standards there are. And I think, again, here the chemists have a leadership to play, because we talk about chemistry, no one else can do it as well as we can. Therefore, we need to show the world what this means, what is sustainability, what is environmentally concerned, and apply it at any scale. You shouldn't hide behind the, it's just a small scale, no one will notice. Take it into account even for the smallest experiments. It's you who decide, you as the chemist, you as the experimentalist. That's it. That's a case story I will talk about after lunch. Are there any questions right now, or you just long for some nice food and rest? You don't need to listen to me for a while. But I'm happy to take a question if there is one or a few now. Yeah, there were. So start with the lady down here. Yes? My personal role?
Yes.
Well, I'm traveling the world talking about these things. So I will go to Milan in a while, I will go to India. There are not so many people with such a long career, if I can call it that, experience talking about these things. So